answering an Edexcel GCSE Biology Paper 1 higher tier from June 21. Figure 1 shows the diagram of a human eye, which row in the table gives the function of structure X and structure Y. So structure X is the cornea, structure Y is the lens. What do they both do? They both refract light so that it arrives here on the retina. So the answer here is D. Describe the changes that occur in the eye when bright light is directed into the eye. So in order to protect the retina from damage, the pupil constricts so that less light reaches the retina, less damage is done to the retina. Eye test can detect some brain tumours. State one other way that brain tumours can be detected, using something like a CT scan, an MRI, X-rays. Describe why a brain tumour is difficult to treat. Think about what's going on. You've got the skull protecting the brain, which you first of all have to get through, meaning that it's difficult to access, and also there's quite a high risk of damage to the brain, because the brain is a very vulnerable organ. It's literally got the texture of set yogurt. Figure 2 shows part of the reflex arc in a spinal cord. Describe how an impulse passes from a relay neuron to the motor neuron. So we're looking here at this gap. It's known as a synapse. So a chemical known as a neurotransmitter diffuses across the synapse and binds to the postsynaptic membrane on the motor neuron. Triggering an electrical impulse. Explain the function of a reflex arc. Well, it protects you from harm, and it is a process that occurs in response to danger. So there's no conscious input from the brain, so you don't have to think about it, which is a good thing because that means it's faster. A scientist investigated the reaction times of 5 students using a computer program. The computer screen showed a blue square at the start. As soon as the blue square turned yellow, each student had to press the key on the keyboard as fast as possible. Figure 3 shows the results for the 5 students, which is the median result for the students. So let's put them in order. 200, 210, 215, 225, 245. I do that to make sure I don't miss anything. What's the middle result? It's 215. The scientists wanted to investigate if the colours of the squares used on the computer program affected the reaction time. The computer program started with blue squares that turn yellow. Describe how the scientists could compare the reaction times of these students when they respond to red squares turning into yellow squares. So in this case, our independent variable, what we're changing is use red squares. The dependent variable, what are we measuring? Measuring the reaction time for noticing that the red squares have turned to yellow. And then we need lots of control variables. So what are we keeping the same? Like the tiredness of the student, their health. And then, as always, repeat and calculate a mean. I can't express enough how important that is. Name the organisation which defines health as a state of complete physical, mental and social well-being, and not merely the absence of disease or infirmity. That is the WHO, the World Health Organization. TB is a communicable disease. State 2 ways that a communicable disease is different from a non-communicable disease. A communicable disease is passed from person to person. And communicable diseases are caused by pathogens. You can keep writing about the fact that non-communicable diseases are the opposite, but actually that's fine. Explain one way that the spread of TB can be reduced or prevented. So we're explaining it. So let's say use vaccination programs. To provide immunity within a population. Because you can't spread a disease if no one's got it. You could say something like avoid close contact. Why? Because TB is an airborne disease. A student researched the number of people with TB in some countries. Figure 4 shows the student's data. Complete the table to show the student's data. So what are our column headings going to be? Looks sensible to say country. And the number of people with TB. So we've got Belgium. Doesn't matter what order we do this. Portugal. I'm crossing them out to make sure I don't forget anything. Explain why people with AIDS are more susceptible to TB. So, AIDS destroys white blood cells, specifically the lymphocytes. So the TB bacteria is no longer killed because you have a very weakened immune system. Streptococcus mutans is one species of bacteria which causes tooth decay. A scientist tested the effectiveness of toothpaste at killing this species of bacteria. The scientist spread the bacteria Streptococcus mutans across an agar jelly plate. Which word describes the techniques used to prevent contamination of the agar jelly? You want a sterile aseptic technique. Give 2 precautions needed to prevent contamination of the agar jelly plate. You want to keep the lid on the Petri dish at all possible times. And use sterile equipment. You could also wear gloves. Work close to a Bunsen burner. A scientist placed a filter paper disc soaked in toothpaste mixed with saliva disc A on one half of the agar jelly plate. A filter paper disc soaked in only saliva disc B was placed on the other half of the agar jelly plate. The agar jelly plate was kept at 37 degrees for 24 hours. Figure 5 shows the results. The diameter of the zone of inhibition around disc A was 9 millimeters. Calculate the area of this zone of inhibition using the equation pi r squared. Use 3.14 for pi and give your answer to 1 decimal place. So the area equals pi r squared. If this is the diameter, then 4.5 is the radius. We've been told to use 3.14 as pi. Put that into your calculator to get 63.585. Then we need our answer to 1 decimal place. Give a reason why disc B, soaked in only saliva, was included in this test to act as a control. Suggest 2 reasons why this test does not prove that toothpaste kills all bacteria on the teeth. Well, first of all, they only used 1 species of bacteria. And also the conditions in the mouth are going to be very different. Like it says on teeth, but we don't have any teeth here. Before new toothpaste can be sold, the toothpaste has to be tested on cultured human cells. Cultured human cells were placed in 4 identical flasks. Saliva was added to each flask and a different brand of toothpaste was added to each of the 3 flasks. The human cells were left for 2 hours and the percentage of cells that were healthy were recorded. Explain the conclusion that can be made from this data. You can see, look, how healthy all our cells are. And if you compare it to saliva only, first of all, the toothpaste has no effect on healthy cells, so it's not harmful to them. And why can we see that? Because the percentage of healthy cells after 2 hours was very similar to that of saliva. Figure 7 shows cells from an onion root tip as seen using a light microscope. Identify the stages of mitosis shown in cell Q and cell R. Remember to use the mnemonic or use PMAT to help you remember. It goes prophase, metaphase, anaphase, telophase. Here you've got the chromosomes lining up at the equator of the cell, which is metaphase. And then over here you've got your 2 nuclei moving to opposite ends of the cell. We're going to end up with the cytoplasm being split down there. This is known as anaphase. So the answer here is A. Describe 2 processes that occur in cells during prophase. So that's the first stage of mitosis. Well, the chromatids condense, so they become more visible. And the nuclear membrane breaks down. State the term used to describe the process which occurs after mitosis when the cell divides into 2. That is known as cytokinesis. The diameter of one cell in figure 7 is 0.075 millimeters. What is this diameter in micrometers? To do this unit conversion, you multiply by 1,000. So B is the answer. A student placed a prepared slide on the stage of a light microscope. Describe how to adjust the microscope to view the slide at a magnification of times 400. The first thing you want to do is use the times 40 objective lens. And then use the focusing wheel to get up to times 400. Arthritis is a condition that occurs when cells and joints get damaged or destroyed. Stem cell therapy can be used to treat arthritis. Discuss the benefits and risks of using stem cell therapy to treat arthritis. So let's start with benefits first of all. So the benefits include the fact that stem cells can be used to replace damaged cells. And why is that? Because they can differentiate. This reduces the symptoms of arthritis, so less pain for the person. What about the risks? The new cells potentially might not function correctly, and there's a risk of side effects. Figure 8 shows how alcohol consumption increases the risk of developing liver cancer. Someone who does not drink alcohol has a 1.0 risk of developing liver cancer. You can see that here. 1 unit of alcohol contains 8 grams of alcohol. Calculate the risk of developing liver cancer for someone who consumes 4 units of alcohol a day. So 4 units is the equivalent of 32 grams of alcohol. Let's look up here. Then read across to the y-axis. We can see the answer there is 1.2 is the risk of developing liver cancer. Describe how cancer develops in the liver. First of all, you need a mutation of DNA, which causes uncontrolled cell division. leading to the formation of a tumour. Hemochromatosis is a disease that occurs when iron accumulates in the liver. A person with hemochromatosis is treated by having 0.5 decimeters cubed of their blood removed each week. This lowers the level of iron in their blood. Give 2 safety precautions needed when blood is removed from this person. First of all, use a sterile needle. You might want to clean the area. You definitely want to wear gloves. Hemochromatosis, I'm not saying that very well, can be inherited. It occurs when a person inherits 2 copies of a recessive allele. So you need that genotype to have the disease. Figure 9 shows the inheritance of this disease in a family. State and explain the genotype of female Z. So up here, we know that the male over here, because of this information, is heterozygous. And then look at some of this offspring. There's a female with the disease. There's one that's a carrier. Here's a male without the disease, and here's a female without the disease. So what must female Z be? Well, because we have this affected female over here, but then we have these unaffected children over here, they must be HH, like that, heterozygous. Why is that? Because the affected offspring must have inherited the recessive allele from her. Whereas the unaffected offspring must have inherited the dominant allele. Hyperion is an extinct genus of horse that lived between approximately 20 million and 0.8 million years ago. Figure 10 shows the skeletons of Hyperion and a modern horse. They do look similar. Give one method that can be used to date a fossil of Hyperion. You want to find a fossil in the rock layer, and then I think the easiest thing here is to use radiometric dating. You could have also said the location in the rock layer or the age of the fossil surrounding it would also enable you to date the fossil. Give one reason why scientists have concluded that the modern horse has evolved from Hyperion. It's because they both have a similar pentadactyl limb structure. Describe how racehorses alive today have been selectively bred to run faster. So you start by humans choose animals that run fast. So that's the favourable trait, and breed them together. Then you choose offspring that can also run fast. Cause them to breed together. And repeat this process over several generations. Discuss the advantages and disadvantages of sexual reproduction and asexual reproduction. So let's start with the advantages of sexual reproduction. Include the fact that it creates genetic variety. Meaning that they're more likely to survive if there's an environmental pressure. Disadvantages mean that you need 2 parents, and also it's much slower, and obviously due to that genetic variation, some organisms may be less well adapted, so more likely to die. So we've talked about sexual reproduction. Now let's talk about asexual reproduction. First of all, as you'd expect, no need to find a mate. There's a rapid production cycle. Organisms are genetically identical, which is good if that's a favourable characteristic that they have. However, the disadvantages are, as you'd expect, no genetic variation. So if there is a selection pressure, they're more likely to die out. But I've said way too much, really. We've got those 6 marks. Chymosin, probably pronouncing that wrong, is an enzyme that causes milk to curdle. When milk curdles, the proteins in the milk clump together and become solid. As part of an investigation, milk was heated in test tubes to different temperatures using a Bunsen burner. 2 drops of chymosin solution were added to each test tube, and the time taken for the milk to curdle was recorded. Which variables need to be kept constant in this investigation? So just so we know, the independent variable, what we're changing is the temperature. So we need to keep other factors the same. We're just going to remove those because we know that they're changing. So what makes more sense, the volume of milk and time, or the volume of milk and concentration of chymosin? Well, we're measuring the time taken. So that's actually your dependent variable. It's not a control variable. So therefore the answer here is C. Explain why the time taken for the milk to curdle decreases from 30 degrees to 40 degrees. Hopefully you recognise that 40 degrees is close to an optimum temperature for enzymes, meaning that there's a faster rate of reaction, and that's because there's greater kinetic energy between the enzyme and the substrate. So more enzyme-substrate complexes are formed because of the higher rate of reaction. Explain what the expected result would be if 2 drops of chymosin were added to the milk at 70 degrees in the test tube. 70 degrees is too hot. The enzyme's going to denature, and therefore you wouldn't expect the curdling to actually happen. It would take much longer or not at all. As part of this investigation, test tubes containing only milk were heated to each temperature and no chymosin solution was added. State why these test tubes containing only milk were used to act as a control. Describe 2 improvements that could be made to this method so that the optimum temperature for chymosin can be found. You want to use a greater range of temperatures for sure. And use a smaller interval between the temperatures, because then you can find that optimum temperature far more accurately. You could also said repeat the test at each temperature. Chymosin can be produced by genetically modifying bacteria. Figure 12 shows a bacterial cell. Explain how to genetically modify a bacterial cell to produce chymosin. So you want to cut open the plasmid, which is this thing here, and chymosin gene using the same restriction enzyme. Make sure you specify it's the same. Insert the gene into the plasmid using a ligase enzyme. Remember, use sticky ends to seal these 2 things together, and that they must be complementary. And then once that plasmid has that chymosin gene, it's known as a recombinant plasmid, and then it's reinserted back into the bacterial cell. Always write nice full answers here. Don't repeat yourself, but fill your answers with scientific detail. Figure 13 shows a sperm cell. Describe how structure A and structure B enable fertilization. So structure A contains lots of mitochondria, which release the energy needed to help the sperm swim. Structure B, that's the acrosome, and that contains digestive enzymes to help digest the egg cell membrane. Figure 14 shows a human egg cell magnified by 700 times. Calculate the actual width of the region indicated by the line on figure 14. Give your answers in millimetres and in standard form. So you're using this formula triangle IAM, which states the actual width is image length divided by magnification. So start by measuring the length of this line here. You'll find it is 4.5 centimetres. We need to get that into millimetres by timesing by 10. So we're doing 45 divided by the magnification, which we've been told is 700. And then once you've done that, you get 0.0643. They want the answer in standard form, however. So that's 6.4 times 10 to the, count it back, 2 minus 2. A student investigated the movement of water. Hen's eggs were placed in vinegar for 2 days to dissolve the shell. This makes the eggs permeable to water. The eggs were then weighed and placed in different solutions. After 24 hours, the eggs were weighed again. Figure 15 shows the results. Evaluate the results of this investigation, include calculations. Right, so let's do a percentage change in mass. Remember, percentage change is done by doing change over original times by 100. We can see we have an increase in mass of 7 for tap water. So we do 7 divided by 77 times by 100. So it's gone up by 9.1%. This one is unchanged. This one's gone down by 2. So we do 2 divided by 77 times by 100. So it's gone down minus 2.6%. So let's talk about what's happened. So egg placed in tap water increased in mass by 9.1%. Why? Because water has entered by osmosis from an area of high water concentration, which in this case is the tap water, to an area of low water concentration, which is inside the egg, across a partially permeable membrane. So we're getting lots of science in there. Now more calculations. Egg placed in 5% salt saw a 0% increase in mass. Why? The solution must have been isotonic, which means it had the same concentration of water. And then rounding it off with what's happening in that final solution. Had a 2.6% decrease in mass because water left the egg. I'm really pleased with this answer. It's very full. It's full of calculations and scientific background. 10a, one gene can have several different alleles. Explain why these alleles can produce proteins with different structures. That's because the alleles have different DNA sequences, which means that the mRNA strand produced has different codons and therefore codes for a different amino acid order. Monoclonal antibodies are proteins. Figure 16 shows the process of monoclonal antibody production. So we have an antigen being injected into the mouse. The lymphocytes of the mouse produce antibodies that are complementary to that antigen. And then we form a hybridoma, which, remember, is a combination between a lymphocyte and a tumour cell or a cancer cell. And why do we do that? Because that cancer cell will replicate extremely quickly, producing large numbers of monoclonal antibodies. Which type of cell is added to B lymphocytes to produce the hybridoma, the hybrid cell? It's the cancer cell. A person with blood group A has the A antigen on the surface of their red blood cells. Monoclonal antibodies can be used to detect the A antigen on red blood cells to determine the blood group of a person. Explain why monoclonal antibodies used to detect blood group A would not react with the blood of a person with blood group B. That's because the shape of an antibody is complementary to the A antigen. Now, the B antigen has a different structure, and therefore this antibody will only bind to the A antigen. It will not bind to the B antigen. The allele for blood group A and the allele for blood group B is codominant. Gregor Mendel investigated the inheritance of alleles using flowering plants. He showed that the allele for red flowers is codominant with the allele for white flowers, meaning that a heterozygous plant, so IRIW, produces pink flowers. Give the genotype for a plant producing white flowers. It needs to be homozygous, so we need two IWs here. Explain the outcome. If two plants that produce pink flowers are crossed, you should complete the Punnett square. So we're effectively crossing two of these. Be so careful with this. Read the question really carefully. And it's just a matter of working out the outcomes. So we have 25%, which are IR, so red flowers. Really take the time to link these phenotypes and genotypes. 25% are IWIW, so white flowers. And then finally 50% are IWIR, which is pink flowers. And by doing all that, I know I've guaranteed myself 4 marks.